Now let's take a look at cells. The cell is the structural and functional unit of life in the hierarchy. Recall from the previous lecture that the cell can perform all the functions necessary to meet that life definition. Now, there is a reason why cells are always small or thin. A cell has to move nutrients and waste across its surface to stay alive. Also, as a cell gets larger, its metabolic needs also get larger. This means that a huge cell would have to have a huge surface to volume ratio to move enough nutrients and waste to stay alive. This combination of huge volume and huge surface is physically impossible. In fact, as the cell gets larger in volume, its surface area decreases relative to that volume. Even if we wanted to create a huge cell, it would never move enough nutrients and waste across its surface to stay alive. In the world as we know it, this limitation of surface to volume ratio results in cells that are either small or long and skinny. In multicelled organisms, specific cell populations tend to take on specific shapes and functions. This specialization allows for increasing complexity in organisms. For example, the cells responsible for detecting light in the retina of your eye are physically and functionally different from the cells that absorb nutrients in your small intestine. Although they contain the same genetic information, this process of specializing will occur during embryonic development for many organs in our body. You'll hear of specialization examples throughout both courses. Organisms must maintain homeostasis at every level of the hierarchy. For multicellular organisms, that requires communication of cells. As a result, every cell in the body will have mechanisms in place for communicating with and responding to other cells and molecules. For example, you have cells in the immune system that can recognize foreign cells by looking at molecules on the surface. If the molecules on that cell surface match your molecules, then the immune system cells consider that a part of your body. However, what do you think would happen with the donated cells? For example, a patient receives a liver transplant. The new liver would have different surface molecules on its cells. The immune system will see those different molecules and attack the liver and reject it. This is why many organ donation patients must take immunosuppressant drugs to prevent that rejection. This is a graphic example of specialized cells throughout the body. You can see how the shapes are very different. Our structure and function theme tells us that the functions will also be very different. Regardless of the type of cell we see, most cells have the same basic components. All cells in your body have a plasma membrane, also called a cell membrane. The plasma membrane serves as a physical boundary, but also performs nutrient and waste transport and contains molecules for communication. All cells contain cytosol and cytoplasm. Cytoplasm is the sum total of cytosol, or fluid, and everything dissolved into it. The cytoplasm will contain organelles, structural proteins, enzymes, metabolic chemical reactions. Most cells of the body contain nuclei. This membrane-bound organelle contains the DNA, which serves as the user's manual for the cell. Everything a cell does, when it starts and stops, how much it does, is controlled by the DNA. The nucleus will also contain the RNA, which is made from the DNA. Note, not all cells are nucleated. Your erythrocytes, or red blood cells, do not contain nuclei and therefore have a short lifespan. The plasma membrane has a unique structure that, as you would suspect by now, determines functions. Let's take a few minutes to discuss the structure. By the way, there's a great YouTube video on Blackboard to check. Structurally, the plasma membrane is a phospholipid bilayer that contains cholesterol as well as integral and peripheral proteins. Functionally, this organelle is a semi-permeable boundary for the cell. So what does that mean? Well, let's take it one molecule at a time. Phospholipids form the backbone of the cell membrane. Each molecule contains three parts: a glycerol, polar phosphate head, and nonpolar fatty acid tail. Glycerol is a three-carbon chain that we will attach the other components to. It's not represented on the slide. Since we have three carbons, we can attach three things to this chain: one phosphate head, two fatty acid tails. Phosphate functional groups are extremely polar or charged. You can think of them as a little negative group. Because they're polar, they attract other polar molecules like water. Therefore, we refer to the phosphate head as being hydrophilic, which translates to water-loving. The phosphate heads are oriented to the outer edge of the double layer. Lipid tails attach to each of the remaining two carbons on glycerol. Lipids are nonpolar or not charged and would therefore repel water. We refer to lipids as being hydrophobic, which translates to water-fearing. As a result, the inside of the membrane prevents polar molecules from crossing. Cholesterol is substituted for the polar phosphate head every now and then. They are represented in the main diagram as yellow strings of hexagons. I've placed a red circle around one of them. Glycolipids. These are fatty acid tails with glucose chains attached instead of the phosphate head. They hold the membrane together and serve as signaling molecules for cell-to-cell communication. How does all of this work together? The phosphate heads will attract polar molecules like glucose and amino acids. However, if that molecule tries to cross the membrane, it is blocked by the non-polar lipid interior. This means that polar molecules cannot cross the membrane on their own and are impermeable. The only exception to this rule is water, which sneaks across the membrane and can carry ions with it. Regarding small lipid molecules, well, they actually can cross the membrane on their own. A small lipid will dissolve across the cholesterol through the lipid interior and exit the other side onto another cholesterol. Again, functionally, what does this mean? The membrane has selective permeability. Because some molecules can cross the membrane on their own, like small lipids and water. But other molecules cannot cross the membrane on their own, like polar molecules larger than water. Also, once a phospholipid bilayer is formed, it's flexible, movable, and easily changed. A cell can pull a piece out or stick a new piece in easily. You can also house larger proteins inside this bilayer structure. With regard to those proteins we find inside the membrane, let's take a look at those on the next slide. Integral proteins. These are firmly attached to the bilayer and serve as transport molecules. Think of them as bridges across the bilayer that we can use to move those non-permeable polar molecules, or as things that allow cells to talk to each other. We have channel proteins. Imagine a tunnel through the bilayer. Anything that fits flows through. Carriers. These proteins transport a specific molecule across the membrane. Receptors, those serve as signal molecules. Peripheral proteins. These do not span the bilayer, but they're loosely attached to one side or the other. Some peripheral proteins act as enzymes, catalyzing a chemical reaction as it is transported across the membrane. Others allow for cell movement, while others are either structural for the cell or link cells together. Glycoproteins. These can form an outer layer called a glycocalyx. These proteins with glucose chains attached are sticky and can serve as signaling molecules for cell-to-cell communication. Here we have an example of a protein type along with its function. The transport is a channel or a carrier. The receptor, cell signaling specificity. Recognition for cell-to-cell communication. An enzyme forming a chemical reaction. An anchor, some kind of peripheral protein attaching organelles or giving shape. And adhesion, linking proteins between cells. All membrane-bound organelles inside a cell will have the same phospholipid bilayer with cholesterol, integral, and peripheral proteins. There are a few organelles that are not membrane-bound. We're going to take a quick look at the function of these listed organelles. The nucleus, that contains DNA and RNA and is responsible for control of all cell processes. Mitochondria, these make chemical energy in the form of ATP. Ribosomes are not membrane-bound, but make proteins. Rough endoplasmic reticulum, RER. Ribosomes are attached. This organelle contains the proteins made by the ribosomes. Smooth endoplasmic reticulum, SER, makes non-protein molecules. Golgi modifies, repackages, and secretes newly made proteins. Vesicles, small membrane-bound organelles that perform specific functions. Lysosomes contain enzymes that break down molecules. A white blood cell that eats bacteria would use these to kill the bacteria. Peroxisomes contain enzymes that neutralize toxic byproducts of metabolism. Microvilli, these are extensions of the plasma membrane that increase surface area. In addition to these organelles, cells may have protein-based non-membranous organelles for support and movement. Microtubules are proteins that form small tubes. These are structural and give shape to the cell itself as well as cilia and flagella. Motor proteins. These molecules move some type of cargo throughout the cell. Microfilaments are two main types, myosin and actin. They also help move things in the cell like flagella and cilia. Centrioles, made of microtubules, these structures form the foundation of the cell's spindle. Cilia, made of microtubules and moved by microfilaments, these long structures move fluids along the surface. Flagella, also made of microtubules and moved by microfilaments. They are specifically used for cell propulsion or swimming. In order for cells to form tissues, we have to stick the cells together to move up our hierarchy. We generally have three types of connections between cells. Tight junctions are impermeable connections between cells. The stomach will have these on its surface. These connections prevent stomach acid from moving between the surface cells to the tissues underneath. We also see this in the skin. Gap junctions are permeable connections between cells that allow the cells to share cytoplasm. The muscle tissue in your heart contains these organelles as well as smooth muscle cells. This allows these muscle cells to share cytoplasm and contract all at once. Desmosomes. These are flexible connections with high tensile strength. We also see these in skin and the heart. Anytime you have an organ that moves a lot, and you need a stretchy and flexible connection that does not easily tear. Now let's see how cells transport nutrients and waste. There are two categories for cellular transport. Passive transport occurs whenever a cell moves something from high concentration to low concentration. The fancy way of saying high to low is to say the cell is moving a molecule with the gradient. This is the natural tendency of molecules to move via Brownian motion anyway, so the cell just needs to let it happen. As a result, a cell is not using chemical energy to move the molecule, and there are several forms of passive transport. Active transport occurs when a cell moves something from low to high or against the gradient. Molecules do not want to do this, so a cell will have to spend chemical energy to force this type of transport. Energy is also used when a cell moves a lot of something at once, or it moves molecules so large that there's not a transport protein in the world big enough to get the job done. Again, there are several forms of active transport. Passive transport diffusion. When a molecule is permeable across the phospholipid bilayer, it will essentially dissolve across the membrane with the gradient from high to low concentration. The cell does not force this type of movement, nor does it need to build any bridges for this. Molecules that diffuse across the membrane with the gradient are small lipids and water. But note, when water diffuses across the membrane, we'll call that osmosis. Facilitated diffusion. Again, we will see a molecule moving with the gradient across the membrane. However, this time, the molecule is polar and impermeable, like glucose. To successfully move the molecule, a cell will build a channel or carrier protein in the membrane. You can think of facilitated diffusion as helped diffusion. It's still diffusion, but the molecules are moving on channels and carriers. This actually allows a cell to move specific molecules. If a cell wants to move glucose, it builds the protein bridge. Osmosis, a special case of diffusion. It is the movement of solvent, in this case water, across a semi-permeable membrane. Not only is water soluble across the membrane, but there's a special channel protein for water called aquaporins, which translates to water hole. We'll come back to osmosis later. Filtration. Again, molecules moving across the membrane with the gradient. This time, instead of the chemical gradient being the key factor, it is a pressure gradient that drives the movement. Therefore, filtration is the movement of fluids and molecules from high pressure to low pressure. Use this figure to practice the transport types. Which of these is osmosis? Which is simple diffusion? Which is facilitated diffusion? Which is a channel? Which is a specific carrier? In active transport, we are moving large molecules or going against gradients. To force this to happen, the cell must use chemical energy. Protein pumps are special integral proteins that use a fuel source to force something across the membrane. Oftentimes, it is moving the molecule against the gradient. Some proteins use ATP directly, where others may use a different energy source. The difference in the energy source is described in the terms primary versus secondary. In the inset picture, you can see two terms that apply to protein pumps. Symport proteins will move two different molecules in the same direction across the membrane. Sodium and glucose co-transport this way in the urinary system. Antiport occurs when two different molecules move in opposite directions across the membrane. Sodium and potassium transport this way in the nervous system. Vesicular transport. Anytime a cell uses a piece of phospholipid bilayer to move something. There are different types depending on the item being moved and the mechanism. In our plasma membrane conversation, we talked about the nature of the membrane and that we can add or subtract pieces easily. Pinocytosis, pinching in of a vesicle from the membrane to transport fluid and small particles. The word transports to cell drinking. An oocyte or human egg cell uses this method to absorb nutrients from the surrounding yolk. Phagocytosis, the pinching in of a vesicle from the membrane to transport a large molecule or particle. This word translates to cell eating. A leukocyte eating a bacterial cell would fall into this category. Receptor-mediated endocytosis, the pinching in of a vesicle from the membrane to transport a molecule that first bound to a receptor on the membrane. Some hormones bind to cells using this mechanism. Exocytosis, using a vesicle from within the cell to secrete a molecule outside the cell. Neurons use this to release chemical messengers called neurotransmitters. Use this slide to review. Use this slide to review. Many factors determine the mechanism of transport. For molecule size, small molecules can be moved with protein pumps, channels, or carriers. However, large molecules must be moved with vesicular transport. For temperature, the warmer the environment, the faster transport occurs. Obviously, there's a limit here. Channels and carriers, these must be made properly by the cell and inserted in the membrane. They must also be available for use. If we were to make a molecule that binds to and blocks a channel, it wouldn't work even though it's there. This is how heart medications called calcium channel blockers actually work. Particle charges, remember polarity? Small nonpolar molecules diffuse directly across the membrane, so simple diffusion works here. However, polar molecules are impermeable and must be moved with facilitated diffusion, protein pumps, or vesicular transport. And concentration gradients, this is the energy source for passive transport. If a molecule must go against the concentration gradient, it must be moved with active transport. Now, as promised, we go back to osmosis. Let's get some terminology out of the way first. A solution is what we get whenever we have solutes dissolved into solvents. In the cell, water is the solvent, and all the other molecules are the solute. You can think of solvent as water and solute as stuff. Cells always start out with the 0.9% solute or stuff and the 99.1% solvent or water. When you add that together, you get 100% solution. Osmolarity. This is the concentration of solute or stuff. The more stuff there is, the higher the osmolarity. It is the osmolarity that determines water movement in or out of the cell. Water moves toward higher osmolarity. A less fancy way of saying that is water moves toward more stuff. Tonicity. This term describes how water moves in response to the amount of stuff. Now note, there are two ways to think about osmosis. So choose one method and stick with it. Method 1, water moves toward higher osmolarity. Or 2, water follows its gradient. I'll use both in each example. Here we have a yellow box representing some environment and a green oval representing our cell. The cell is in homeostasis with a 0.9% solute or stuff and 99.1% solvent or water concentration. Now imagine we put that cell in an environment with the same concentrations of stuff and water. Now, method 1. Is there a difference in the amount of stuff on either side of the cell membrane? Nope. Or method 2. Is there a concentration gradient for the water here? Well, nope. So where will the water go? Well, there's no net movement of water here because there's no gradient to push the water flow. For every one water molecule that enters the cell, exactly one water molecule will exit the cell. Water movement here will be balanced, and the cell will be in homeostasis and will stay the same over time. We would characterize this environment as being isotonic. That kind of means same stuff. So in lay terms, whenever you put a cell in an environment with the same amount of stuff as the cell, there'll be no net movement of water. So follow-up question. Think about organs and organ systems that might help your body maintain homeostasis with osmolarity. Can you guess them? The same setup with regard to our cell, 99.1% water and 0.9% stuff. Now let's put the cell in an environment that is 1.5% stuff and 98.5% water. Method 1. Is there a difference in the amount of stuff on either side of the cell membrane? Yes, there's more stuff outside the cell. Method 2. Is there a concentration gradient for the water here? Yes, there's more water inside the cell. Where will the water go? No matter which method you're using, water will leave the cell and go toward the environment. Over time, the cell will lose water to the environment and shrink. We would say that this cell will crenate. Crenation can be irreversible because not only does water leave the cytoplasm, but it also leaves tiny spaces in molecules and proteins. This is more than enough change to kill a cell quickly. We would characterize this environment as being hypertonic. This term tells us that the environment has more stuff than the cell. So in lay terms, whenever you have a cell in an environment with more stuff as a cell, there will be a net movement of water out and the cell will crenate over time. Same setup with regard to our cell, 99.1% water and 0.9% stuff. Now let's put this cell in an environment that is 0% stuff and 100% water. Method 1. Is there a difference in the amount of stuff on either side of the cell membrane? Yes, there's more stuff inside the cell. Method 2. Is there a concentration gradient for the water here? Yes, there's more water outside the cell. Where will the water go? No matter which method you're using, water will enter this cell. Since this is an animal cell with no cell wall, the movement of water in cannot be controlled. This is more than enough gradient for the cell to swell and explode. We would say that this cell will lyse over time. In fact, I've tested this scenario and it happens in a few seconds. We would characterize this environment as being hypotonic. This term tells us that the environment has less stuff than the cell. So in lay terms, whenever you have a cell in an environment with less stuff as the cell, there will be no net movement of water in, there will be net movement of water in, and the cell will lyse over time. These are scanning electron micrographs and diagrams of erythrocytes or red blood cells in the three environments. Osmosis is a big deal, and it is an important part of many pathologies. You really need to explain, diagram, and predict this concept, and I'll test you on it more than once. Osmosis is behind pathologies experienced by diabetics. In a diabetic, blood glucose levels are higher than normal. Do you remember our homeostasis discussion? Glucose is sticky, so where glucose goes, water will follow. The intracellular fluid and plasma of a diabetic are essentially hypertonic, more stuff. Using what you have learned about osmosis, use this diagram to predict water movement between intracellular and extracellular fluid. Water follows its gradient. Water moves toward more stuff. Now, using the internet, look up the terms polydipsia and polyuria. Then, use what you've learned about osmosis to explain these two symptoms in terms of osmosis and elevated blood glucose. The next slide contains questions to guide you. What does this fluid flow do to blood volume? What does this fluid flow do to blood pressure? What does this pressure change do to filtration in the kidney? What does this filtration change do to urine output? And if blood glucose remains high, what will eventually happen to blood volume? Again, you should be able to diagram and explain the answer to each of these questions to a classmate on the discussion board. Our course focuses on normal physiology. However, for you to be a great healthcare provider requires that you master pathophysiology. In order to do that, you must first master normal physiology. So I'll be giving you a lot of examples and many case studies in pathology, especially diabetes related. These are unfortunately common pathologies experienced over time by diabetics that do not keep blood glucose levels under control. Is this worth a Snickers bar or a mocha latte, frappe, unicorn, whatever every day? Now, before you think I'm judging you, you should know that I found out a couple of years ago that I developed prediabetes. That was after a significant weight loss. I immediately stopped eating all forms of sugar, grains, and starches because I refused to allow myself to develop type 2 diabetes. And no, the body does not require carbohydrates in the diet. That's a horrible fallacy and it's not backed by science. Your liver can make glucose from fats and proteins if it needs to. But sadly, the Western diet of processed food is carbohydrate heavy. It's so much so that it's causing an epidemic of metabolic syndrome, prediabetes, and type 2 diabetes in the United States. In the last few years, public health officials are seeing the same trends in other countries. Prevention of diabetes begins with daily diet. You can see the dramatic rise in rates in the late 90s. It looks like it might start dropping off, but only time will tell. You will see standard of care and dietary recommendations from the American Diabetes Association probably change in the next few years, as it is currently under review. Will I test you on these numbers? No. Should they impact your train of thought and how you eat? Yes. Keep this in mind when you become a healthcare provider, because you will have more and more patients with diabetes needing special care. Now, diabetes starts with metabolic syndrome. This is a disruption in the homeostatic mechanisms of our endocrine system when it comes to insulin and its effect on our body cells. Before diabetes even starts, changes in blood lipids occur. There's often a decrease in good HDL cholesterol and an alarming rise in triglycerides. Because the body cells are constantly exposed to insulin because of diet, those cells start to lose their sensitivity to insulin and stop responding to it. This is called insulin resistance, and it is the beginning of diabetes. So how does insulin resistance start? With diet. You eat too many sugars and starches, and that causes your blood glucose to rise. Your pancreas secretes insulin, and your body cells respond by taking the glucose. That causes blood glucose to drop. It's an endocrine system, negative feedback homeostasis here. However, when this happens too often, the body cells stop making insulin receptors. The cells will now resist insulin, and glucose stays in the blood supply to be converted to fat and stored by our fat cells. Before too long, glucose starts to stay in the blood supply too long. Your pancreas might try secreting even more insulin, and the resistance continues to build. So glucose stays in the blood supply too long. You see the trend here, right? The only solution at this point is to break this cycle by changing diet dramatically. Here's another example of the importance of osmosis. When the liver begins to fail, the liver cells stop making proteins we find in our blood. Those proteins are important for osmosis at the capillaries because they create an osmotic force at the capillary. This osmotic force pulls water from the interstitial space back into the blood vessels. During liver failure, that protein's no longer made. Now, there is no osmotic force to pull water into blood vessels. That water will now stay in the interstitial space and cause horrible edema or swelling. People can even develop blisters from fluid that is collected in the interstitial space. I've been told by clinical faculty that they'll find a liver failure patient early on in the clinical setting. They'll bring students to this patient and ask the student to explain the relationship between liver failure and the edema. So make me proud and tell them. Failure to produce plasma proteins by the liver has disrupted the capillary osmotic pressure. Fluid is left the capillary by filtration but cannot be reabsorbed due to the drop in osmotic force. Or something like that. You'll learn the specific words I just used in AMP2. If you're panicking, remember, you can pull the transcript of this lecture and write those words down specifically.